I'm Lucy Martel, and my poster is on Bacillus anthracis and what makes it so special. So I was really interested in the anthrax poisoning cases that have come up lately, and I really wanted to see what made that so favorable for bioterrorism. And it's known that it's part of the Bacillus cereus group and Bacillus thuringiensis. The three of them all share very similar physical and genetic compositions. So I really wanted to see what made Bacillus anthracis so different. So as you can see in figure 1, this is Bacillus anthracis, this is Bacillus thuringiensis, and this is Bacillus cereus, so they all have the same kind of size and shape, and they produce the same type of spore and that kind of thing. So I really wanted to see what made the anthracis so different, and my research led me to find these 3 genes, the edema factor, the lethal antigen, and the protective antigen, and these 3 are responsible for the capsule and the toxin that make Bacillus anthracis harmful to humans and animals. And typically in the United States we only get 1 to 2 cases of anthrax poisoning a year, just because it is typically found in soils and in animals and that kind of stuff. But in 2001 there were 22 cases. So basically what I did is compare recently assembled strains of Bacillus anthracis to known reference genomes of both Bacillus anthracis, Bacillus thuringiensis, and Bacillus cereus in order to determine their differences or similarities. So I took some raw data from the NCBI and the ENA, and I ran it through a bunch of different sequences that we call the pipeline in class. And here's a general outline of that pipeline. So we get the data of interest, we download it to our computers, and then we run it through the software that trims off any additives that were used in lab to sequence that data. And then we can chop it up into small pieces so that we can read it and then make that into one larger piece and then compare that to a reference genome of my choosing. I chose the Ames strain, which was used in 2001, which was the Amerithrax strain, for the vast majority of my project. And here's just the species names that I used, links that I got them, and where they were found. They're found in a majority of different things. Most of them were soil, sheep, cattle. One of them was a zebra, and then some of them were just mutant strains that were found in the lab. But I thought those would be interesting to kind of compare it to. So one of the steps we run is called QUAST, and that compares the recently assembled strain to the strain. Like I said earlier, I referred it to the Ames strain. And you get a figure like this from that report, and it just shows how similar or how aligned it is to the reference genome. And here the red indicates where it's poorly aligned, and the green is where it's aligned really well. And with most of the strains that I was looking at, there was a vast range of how similar or how dissimilar it was to the Ames strain, which I thought was really interesting because they're all the same species. So then at the end of it all, you can run all of your strains through a program called Rory and get a phylogenetic tree, which I have right here, and it just shows how related the strains are to one another. And you can see that the Bacillus anthracis is all very closely related, while the Bacillus cereus and thuringiensis, which is the gray and orange respectively, are kind of mingled in with one another, and it's hard to decipher where the 2 species are completely different. And I also included a student here in the microbiology class, Rachel Graves, her data, because she was trying to decipher which strain her data belonged to, so I threw that in with mine. And even though it wasn't concrete about which strain it was, it's definitely not anthracis, which was something that helped her in her poster. So then going back to the 3 genes that I was looking at, I decided to do a presence-absence table, and I looked for the 3 genes in all of the data that I had. And I just looked for them in the software, and a plus indicated if it was present, and a minus indicated if it wasn't. And for the most part, unless it was a mutant strain of Bacillus anthracis, they were all present, which was interesting because none of them were present in thuringiensis or cereus. So that's really interesting, I guess. So then I compared the average nucleotide identity between a number of different anthracis genes, or strains rather, and 2, well, one of each, one Bacillus thuringiensis and one Bacillus cereus, and I just wanted to see how similar they were to one another. And here the green indicates a value that's acceptable, and that the black is not acceptable. And basically what you can see here is that the Bacillus thuringiensis and cereus are very closely related to one another, but they're not close enough to Bacillus anthracis, and vice versa for the other ones. So basically, I was just able to determine that Bacillus anthracis is relatively different from Bacillus thuringiensis and Bacillus cereus, but those two latter ones are very closely related.